signal, because EO2, P10, because if we get the stability.
Well, again, thanks very much for the invitation. It's been a really fun day. It's always great when you have a chance to talk to the students, and I really enjoyed that and talking to many of the faculty here. And been very impressed by the quality and the approach that people are taking to science here at a time when I think we all hope that the great advances that are being made in genetics and other areas really translate into changes in human health. I think places like this are really going to be places that make a big difference, and it's really been great to be here. So what I'm going to try to do today is to talk about our work on regulatory T cells. So I'm going to be going through a few different areas. We'll see if I make it. I may not. I've got to shorten my CV, clearly, to give me more time to give my talks. But I think that hopefully you'll get some idea of some of the things that we're trying to do in the lab these days, in the hopes that we can give you some insights into what we're working on. So the first thing I need to do is to tell you some disclosures of some of the things that we're doing in case they come up during the talk. We get some support from Becton Dickinson for some of our regulatory T cell studies, the clinical studies in particular, where we use clinical grade antibodies for cell selection. As mentioned, we're working on an anti-CD3 antibody, which is being developed by a company in Maryland. And finally, I'm on the scientific advisory board of a company interested in regulatory T cell therapy. So when I think about the immune system and how it works these days, it sort of comes down to one slide for me. And each of these areas, I think, are really quite vital to thinking about how we're going to induce tolerance in cases where we don't want an immune response to work, or immunity in cases where we do. And I think it's a combination of genetic predisposition, an immune system that's either focused on being able to give a positive immune response because of polymorphisms that exist in these genes, or to suppress an immune response depending on the alleles that people express. But in addition to these critical genes, and there are many more of them involved in central and peripheral tolerance, there are clearly environmental factors, broadly speaking, anything from the chemicals and bacteria and viruses in our environment to the commensals that live in our body, that together help to influence the balance from one which is pro-regulation based on a variety of cell types that exist to pro-immunity based on other cell types. And it's really creating a balanced environment, an immune homeostasis, which I think is at the core of what the immune system is designed to do. And when we look at things like infectious responses or autoimmunity, what we're looking at is perturbation of what otherwise would be a homeostatic system. And so when I talk about regulatory T cells, I talk about them in that context, that these are cells really designed to maintain a core homeostatic system for the body. I have competition. So for those of you who have been asleep for the last 15 years, let me just take a second to tell you a little bit about what I mean when I talk about regulatory T cells. So as I've already alluded to, the immune system is a very dynamic system in which naive T cells recognize foreign antigen and make either an effector T cell response or autoantibodies in the case of diabetes, which I study a lot, and that these cells then go on to destroy target tissue. But what multiple investigators have found, really pioneered by Shimon Sakaguchi and Bruce Hall, really was that there was a unique population of cells that are present in the thymus and exported continuously from the thymus, which we call natural regulatory T cells, or natural Tregs, which are defined by a variety of criteria, and really these are the criteria I'm going to use in my talk today. First of all, they're a CD4 positive lymphocyte. They are a cell population that makes FOXP3, a transcription factor that largely controls their suppressive program, and it's a population of cells that accounts for anywhere from 4 to 7% of all of the CD4 positive cells in the blood. And these cells are general homeostatic cells. They're found in tissues ranging from skin, fat, muscle, and of course the lymphoid organs, and they're designed to keep animals in a state of homeostasis. In addition, though, when a naive cell encounters antigens in a certain way, usually in the context of cytokines such as IL-10 and TGF-beta, There's a second regulatory population that has many, if not most, of the same characteristics as the natural Tregs. And these so-called adaptive Tregs are also CD4 positive, also express FOXP3, also have many of the suppressive activities as the natural Tregs, but tend to exist locally during an inflammatory response and are often responding to a different set of antigens than the natural Tregs, because these come from the thymus, where they see self-antigens, and these are coming from a naive repertoire that's selected differently, that are recognizing the antigen that's exposure. And together, these two populations are absolutely key in almost every immune regulatory response that we see in the host. And there are a number of ways to know how important these cells are. One is that if you eliminate FOXP3, both mice and people who have a mutation in the gene die very early in age because of massive lymphopoiesis and infiltration of tissues and destruction. But also, I think, is the plethora of activities or functions that these cells have been purported to have in various systems. And typically, they are very locally acting activities that happen at the site of inflammation or damage. There are larger, more robust activities like active TGF-beta when you get massive inflammation. And there are these sort of general homeostatic control. But all of these functional activities have been documented in one setting or another and largely function to maintain, again, this quiescent immune system. So, of course, one asks the question, when you have such a dynamic, robust, highly specific cell population, why do people get autoimmune disease? And it turns out, if you look very carefully, we now know that in people that have autoimmune disease, and particularly there's lots of data on diabetes, rheumatoid arthritis now, multiple sclerosis, SLE, what you find is a common set of observations, and I'll show you both a summary and some of the data. So the first thing we've noted that in a number of these autoimmune settings that there's an inadequate number of regulatory T cells, and specifically at the site of inflammation, whether that's due to a lack of growth factors like IL-2, co-stimulation like CD28, where these cells absolutely need these two molecules to survive and develop, or whether they're not proliferating well or surviving very well is not always clear. But the bottom line is that at sites of inflammation in people with autoimmunity, these cells are down. The second thing is that not only are the numbers down, but their activity is down. And when you look, you'll see a failed ability to maintain good contact-dependent suppression, reduced amount of cytokine production like IL-10 and TGF-beta, and an overall dysfunction of these cells, again, often at the site of inflammation. And then finally, I think, and most significantly from an adaptive point of view for the immune system, is that during the course of patients' development of autoimmune disease, you see an ongoing increase in effector cells that become resistant to Tregs. And nobody knows really at the moment what characteristics there are about this. We know that cytokines such as IL-6, for instance, will promote this resistance, but they end up being less regulatable and as such more pathogenic. So let me show you an example of that. If you take the mouse model of type 1 diabetes, the NOD mouse, and you look at this animal over time in the islets themselves that are being inflamed, what you see is over time a direct correlation between the number of regulatory cells in the tissue as a consequence of the overall number of CD4s. That as the islets get more inflamed, there are less regulatory T cells to effector T cells. And it's not just that the effector cells have grown out of control, there's an absolute loss of Tregs. And you can see that here when you look at expression of key molecules, FOXP3 and CD25, the IL-2 receptor, that the islet resident Tregs are far less effectively able to respond to growth factors like CD20, like IL-2, and you can see the mean fluorescence of the receptor here versus those cells that are coming out of the draining lymph node that's right outside the pancreas itself. And so these cells go on to die, and I'll show you they also change their phenotype and actually change their function. So how do we know that they not only died, but they changed their function? We made a reporter mouse that not only lit up Tregs because they were expressing FOXP3, but also permanently marked the Tregs that at some point had expressed FOXP3. So a true Treg is both green, because it's driving FOXP3, green fluorescence protein off of FOXP3, but also yellow, or in this case, actually, this is a red marker, because at some point they did express FOXP3 and turned on this reporter lineage gene. And so if you look in the average NOD islet, what you see are a number of cells expressing FOXP3. They're both green and yellow. Here I'm also stating for FOXP3 protein. But a significant number of cells have lost FOXP3 expression. So you wouldn't be able to tell if it wasn't for this reporter mouse that these cells were even there because they're low for FOXP3. But because of the reporter lineage marker, we can see them. And in fact, if you isolate and look at these cells directly, you can see that a significant percentage of these cells, and in fact some of these cells that are starting to lose FOXP3, are actually starting to make a pathogenic cytokine, gamma interferon. So over time, when cells are losing their FOXP3 expression, losing their IL-2 receptor expression in the islet, and this is true in the brain as well in EAE, a model of multiple sclerosis, you see this not only loss of the regulatory activities, but also the increase in pathogenic activities. And this is not just unique to mouse models. If you look in humans, I'm showing you here again type 1 diabetes, but it's also true in multiple sclerosis and RA. The percentage of regulatory cells that make gamma interferon are significantly increased when compared to healthy controls. Again, reiterating the fact that under autoimmune settings, there is a defect in these cells, and they lose FOXP3 expression, they lose IL-2 receptor expression, and become potentially pathogenic. Here just again shows this gamma interferon producing population here. And interestingly, if you isolate them, these cells are much less effective at suppressing an immune response compared to their gamma interferon negative counterparts from the same individual. So this just is one example. Another example of how these cells are defective in the autoimmune setting is some work that's been done by Jane Buckner's group, now looking at a downstream signaling molecule following IL-2 engagement with the IL-2 receptor, STAT5 phosphorylation. And you can see here, first of all, again, that Tregs from type 1 diabetics suppress less well than Tregs from a healthy age-matched control. Here's a dose-response curve. And interestingly, when you look at the ability of Tregs to respond to IL-2 in these patients, you see a diminution in the phospho-STAT5, the phosphorylation of STAT5 after IL-2 engagement, both here with high IL-2 as well as with a low IL-2, and here's a dose-response curve. So not only are these cells low for IL-2 receptor, but they're able to signal less well the STAT5 molecule. So to summarize then, what we believe happens during an autoimmune response is that over time, as a combination of T effector resistance, Treg loss, and instability and reprogramming, you get this overexpression of an autoimmune response and destruction of your target tissue. So what I'd like to spend the rest of the talk, because that's obviously published and pretty fundamental, is to ask three questions that hopefully I can get to today. One is this instability that we've identified and talked about now for a while. Is this just a general defect in autoimmunity, or does it require, or is it a consequence of antigen exposure? Secondly, what are the biochemical pathways downstream of this that in fact are responsible for some of this instability? And then finally, I'd like to spend the last part of the talk talking about our attempts to try to clinically apply Treg technology for treatment of autoimmune disease and organ transplantation. So let's start out with the Treg instability. Again, we're using the same mouse that I talked about before, but now instead of diabetes, we're looking at EAE. And the reason we're looking at this neurological disease is because we now have tetramers. These are MHC peptide molecules that can be used to bind to and identify the antigen-specific T cells that come up during the course of an immune response. And so we now inject mice with this autoantigen, MOG peptide, 35 to 55 in CFA and pertussis toxin. And then we follow this disease as it comes, and then the animals go into recovery. And this is kind of the pathology of what happens during that process. And we can now monitor what happens to the effector cells and the regulatory cells. And so here, if we did exactly what we did in the NOD mouse, which is to just look polyclonally at all of the Tregs, what you'll see is that the overwhelming majority of the Tregs in the lymph node and spleen of a B6 animal are expressing both FOXP3 and the reporter, and a very, very few percent of them have lost any FOXP3 expression at all. After the immunization with the peptide, we start seeing a small percentage, 10 or 15% or so, which have downregulated FOXP3 in general. And at the peak, that still isn't much more than you might see anywhere else in the animal. So this looks generally like in the NOD. There are some cells, and if we looked at these, they'll make gamma interferon, but it doesn't look much different depending on the course of the disease. But if instead you look at the tetramer positive cells at the same time point, you see something very different. First of all, there aren't a lot of them in the resting healthy animal because we haven't immunized yet with MOG, and the few that are there are FOXP3 positive. But now if you look on day 8, again, in the draining lymph node and spleen, the majority of these cells are both GFP and YFP positive, suggesting they're bona fide Tregs. But now if you look in the CNS, you start seeing now a significant percentage of only these antigen-specific cells that have lost FOXP3, and they're losing it more significantly than in the polyclonal response, both as a percentage. Here you can see that it's almost a quarter, more than a quarter of the cells, where here it's less than 10% of the cells. And then in remission, they go away. So this population here is uniquely present within the autoantigen. And you can actually look at that in different ways by adoptive transfer and see here that if you transfer in FOXP3 negative cells, they don't become FOXP3 positive. But if instead you sort out the antigen-specific cells, you can see that it's really within this antigen-specific pool that majority of these cells have lost FOXP3 expression, not in the polyclonal pool from the same animal at the same time point from the same tissue. So we would conclude then that the antigen-specific response is in part responsible for the instability that happens in the course of autoimmunity locally in the inflammatory response. Now, we don't think that's the only thing that's required. Certainly other cytokines and other factors are probably playing a role, but it's preferentially happening within the antigen-specific population. Why does that matter? Well, it matters for two reasons. One is if those cells start making gamma interferon, you can imagine what it's like to have an antigen-specific, MOG-specific T-cell making gamma interferon or IL-17. That's going to potentially exacerbate the disease, and we can show that you can adoptively transfer these cells, and they will indeed promote disease. But also it suggests, and I'm not going to have time to show it today, is that a lot of these cells, these antigen-specific cells, are also coming from the adaptive immune response, this response that's happening in the course of an inflammatory response. And these cells are just fundamentally less stable. And so they started out trying to become Tregs, probably to cope with inflammation, but they lose FOXP3 and don't keep it on permanently, maybe enough to turn them YFP positive to keep the Cre on, but not long enough to stabilize. And so a lot of these cells here are coming from the adaptive pool. And one way you can look at this is by looking at the methylation state of the FOXP3 gene locus itself. When Tregs are stabilized and have fully developed, they demethylate a region of the FOXP3 locus, and that can be monitored. And so if you look, what you see, if you look at a regular naive cell, the majority of the FOXP3, overwhelming majority of the FOXP3 is methylated. Even in the memory T cells, the majority is methylated. But if you look in these cells that are losing FOXP3 expression, you can see a very significant percent of these cells are still unmethylated, suggesting that at one point they had been fully expressing the FOXP3 locus here. Less so in the GFP, more so in the GFP low than in the GFP negative, which may be the adaptive Tregs here. But the bottom line is that the majority of the cells that we think we're seeing here are cells that at one point had a stabilized FOXP3 expression, but have now lost FOXP3. finally, to close the loop here, can we in fact repair this defect that I've identified both in the NOD as well as in the EAE? And the answer is yes. And you can repair it by treating the animals with IL-2, the growth factor that we believe is essential for maintaining Treg stability. So what we've done here is we've taken mice after immunization, and we've treated it either with PBS. This could happen with the jet lag. Bear with me. PBS, in which case, of course, you still have this third of the cells are FOXP3 negative, or with an IL-2 anti-IL-2 complex, which is very good at activating Tregs. And you can see when you do this that you lose all of these ex-FOXP3 positive cells. So IL-2 can repair the defect. And most importantly, not only do you lose these ex-FOXP3 positive cells, but you can prevent and in diabetes even cure the disease as a consequence of IL-2 treatment. And we'll get back to this a little bit later because actually we've conducted a clinical trial to look at what would happen with IL-2 treatment in a diabetes setting. So to summarize then, we think that autoantigen-specific Tregs are really the predominant population that downregulate FOXP3 at the site of inflammation, that these ex-FOXP3 positive cells in the CNS are highest during the peak response and drop dramatically during resolution, that a subset produce inflammatory cytokines such as gamma interferon and IL-17, and that the majority of the Tregs that downregulate FOXP3 exhibited demethylated TSDR, suggesting that FOXP3 loss occurs in the antigen-specific bona fide Tregs, at least some of them. And that finally IL-2 and anti-IL-2 complexes can in fact be used to stabilize these Tregs. So I've suggested that in fact, as a consequence of inflammation, these cells can become unstable. The question is, what are the downstream targets? I alluded to the fact that STAT5 may be important in the autoimmune setting, as patients with type 1 diabetes or RA or MS have lower phospho-STAT5. But are there other molecules that are involved in this? And so the first suggestion that we might was some studies that were published a few years ago when we eliminated microRNAs and showed that those were absolutely critical for stabilizing Tregs and that without them you lost both FOXP3 expression and the ability to control an immune response. But that was using very broad destruction of microRNAs using deletional models where no microRNAs existed. So we raised the question, could we find a specific microRNA that might be controlling some of these downstream signals? So we did a microRNA array comparing cells that were activated with anti-CD3 and anti-CD28 under stable and unstable conditions. And we found a whole series of microRNAs that were in fact controlled in this setting. And interestingly, many of them fell into the same family, the so-called miR17 to 92 cluster. And so we focused on that pathway. miR17 to 92 had already been known to be very important in the immune system in a variety of studies, so we thought that this would be a hint. And so just to confirm, we took Tregs from mouse and human and confirmed that these microRNAs were selectively overexpressed in this population. Here's doing a dose-response PCR. But most importantly, when we selectively knocked out miR17 to 92 out of Tregs using that same FOXP3 Cree that I've been talking about before, that lineage marking mouse, what we found is that animals in which we had selectively knocked miR17 to 92 out in the Tregs not only didn't go into remission, but had much worse disease than their control counterparts. And this just here shows you the cumulative incidence of the number of mice that actually either died or had the highest EAE scored in the knockout versus the control animal. So where was the miR17 to 92 working? So we took advantage of being able to identify and isolate the antigen-specific cells again, like we had done in earlier studies here, using the MOG tetramer that identified the antigen-specific cells. And what we found was that in non-activated naive cells, the number of Tregs didn't change. Even if you looked at the polyclonal Treg population that didn't bind to the tetramer, the number of Tregs didn't change. But if you looked instead at the antigen-specific Treg population, you saw a dramatic decrease in the number of antigen-specific Tregs that expanded in the knockout animal versus the wild type. And this just quantitates it here to show that the antigen-experienced Tregs were the ones that were particularly sensitive to the miR-17 to 92 locus molecules. So was it a survival problem? Because miR-17 to 92 could be important in survival of the antigen-specific cells during expansion. To test that, we bred the animal to a BIM knockout. This is a very important molecule involved in apoptosis, so we tried to eliminate apoptosis, and we saw that we could in fact recover some of these cells from doing that, but it was not sufficient to control autoimmunity. Here, if you look at the knockout versus the wild type, this is the autoimmunity and the score, and here in the BIM by wild type, by knockout here, you saw that you still had disease in 75% of the animals, and although the score was a little bit lower. So it wasn't just that the miR-17 to 92 was controlling the survival of the cells, it was more. And so when Lucas Jecker in the laboratory studied further, what he found was another major defect in these Tregs, and that is control of production of IL-10. And we know from a variety of studies that Tregs require IL-10 production to control EAE. And so not only does miR-17 to 92 have some effect on signaling leading to cell survival, but its perhaps major effect is in signaling that controls IL-10 production. Again, suggesting that if now we went back, which we've done, and looked at those same cells that are making gamma interferon that I showed you were potentially pathogenic are now making less IL-10. So I think there's a yin-yang there that miR-17 to 92 probably is involved with. So we know that, that there are a number of downstream targets of both TCR and CD28 and the IL-2 receptor that might influence these downstream targets that might lead to changes in BIM, leads to changes in IL-10. But we also knew that one of the major targets for miR-17 to 92 was this protein P10, which controls the expression and the activity of PI3 kinase, which is downstream of both the TCR and of IL-2 receptor. And so we thought that maybe in addition to IL-10 and in addition to BIM, that perhaps activation of PI3 kinase might be involved in the miR-17 to 92 mutation. Why is this such an important target? Well, it turns out that I already told you STAT5 signaling is critical for Tregs, but AKT actually has the opposite effect. Unlike most conventional T cells where AKT activation is essential for activation of T cells, in Tregs, activation of AKT is bad. And anything that would regulate P10 and allow more activation of PI3 kinase could be bad for Tregs. So to test that, whether, and as I said, we knew that miR-17 to 92 was a target. So to test that, we knocked out P10, again under the FOXP3 promoter driving CRE. And one of the things that you can see here, this is a preliminary result, but I think it's real, is that if you look at the constitutive activity of, in this case, AKT in these cells, you can see it's higher in the P10 knockout than it is in the wild type, consistent with what other people have shown, that P10 knockout leads to overactivation of PI3 kinase. So what happens to Tregs in this setting? Well, I know I'm a broken record here, but it's not a surprise. The exact same thing. We now started losing FOXP3 expression and loss of FOXP3 function. We started losing CD25 expression, here shown in a histogram, here shown versus FOXP3. So in fact, loss of P10, overactivation of PI3 kinase gave us that same unstable, non-stable phenotype. Here's doing it in vitro just to show how rapidly this can occur. If you take cells at day 2 after activation, this is now making these adaptive or induced Tregs in vitro with TGF-beta. You can see at day 2 things look good, but by day 5 the P10 knockout is clearly losing both FOXP3 and CD25 expression much more than the control population. And so these acute alterations in signaling after P10 may directly regulate CD25 levels. What's the implication of this for the mouse? Well, you start seeing almost immediately an increase in the number of activated T cells, both CD4 and CD8 T cells based on expression of the activation marker CD44 and loss of CD62L. And most importantly, again, I'm sorry, here comparing the wild type versus the, and most importantly, what you see is these animals get major lymphoproliferative disease and infiltration of a variety of tissues, here showing you kidney, and a majority of them have this lymph node hyperplasia. We've done this work collaboratively with Larry Turka at Harvard. So it appears then that the mere 17 to 92, or at least in general, Tregs really do depend on this balance of positive STAT5 activation, negative AKT activation, and anything that shifts or alters that balance, like a microRNA is likely to do, can have an effect. One final thing that we've done, which may have some clinical application as well, is ask whether we can fix this problem. And the answer is we can. Again, looking at the knockout versus the wild type, the knockout has much more loss of FOXP3 and CD25. But now if you take this animal and these cells and you treat them with a PI3 kinase inhibitor, you can actually restabilize these cells here. Here's using wortmannin to inhibit mTOR and rapamycin. And in all cases, inhibiting downstream signals from PI3 kinase or PI3 kinase activity itself really stabilizes these cells quite a bit. And here, if we, this is again even taking it in real time and looking at what happens even in the stable population if you put them in culture. Again, in the presence of the inhibitor, you see stabilization, you don't lose these markers. So we believe that this, and finally, sorry, a combination then of the PI3 kinase inhibitor and IL-2 is the best and gives you the most stable long-term Tregs. So we believe what's happening is that in fact IL-2 itself is delivering a positive feedback here for STAT5 that controls CD25, but if PI3 kinase is not held in check by P10, that now it counteracts the positive effect of the IL-2 on STAT5. So any application of things like IL-2 are going to require balancing these two pathways so that we don't end up with something where you get activation down PI3 kinase pathway. So this just summarizes where we think we are right now downstream of the IL-2 receptor in CD28, and that is that MER 17 to 92 acts at least 3 places in the pathway. P10 is absolutely essential, and if you overactivate PI3 kinase or underactivate STAT5, you end up with loss of CD25 and loss of FOXP3. So again, going back to the model here, we think that what happens in autoimmunity is there are CD28 IL-2 dependent activation of the effector cells and in fact Treg expansion that happens early on, but that over time you get a defect in these pathways, probably sopping up of the IL-2, maybe loss of CD28 interaction with its ligands, and this resistance loss and instability. And it's molecules like MER 17 to 92, P10, and IL-10, which are critical in these pathways. So I want to spend the last few minutes, how am I time-wise? Am I okay? Okay. I want to spend the last few minutes sort of moving this from a sort of fundamental basic science talks to can we in fact do something in the clinic to try and take advantage of the biology that we've been studying for so many years. Well, I also already alluded to the fact, just want to remind you, that, that if you take animals that are defective or have lost Tregs in the islets, here the FOXP3 level of expression, I had shown you this before, and you treat with IL-2, you can actually enhance Tregs, and you can also prevent disease, and you can even reverse diabetes in this setting once they've already gotten. This is EAE, and you can reverse diabetes. So we thought that IL-2 might be something to try in the clinic. So while I was still director of the ITN, we set up a clinical trial in which we looked to see what would happen if you treated with low doses of IL-2 here. And we did this under the cover of rapamycin, not because we were that smart in terms of stability of Tregs at the time, but because we wanted to protect ourselves just in case the IL-2 activated effector cells by mistake. We wanted to have rapamycin on board to try to prevent the activation of effector cells inadvertently. We treated with the IL-2 for a month, the rapamune for 3 months, and then we followed these patients out. These were new onset diabetes patients who were still making some insulin, some as read out by C-peptide. And what we found, much to our positive surprise but enjoyment, was that you saw a significant increase in the number of Tregs here within the first 28 days in all 9 patients. You saw a significant increase in the absolute number. It wasn't just the percent, but actually the absolute number of Tregs that came up in the peripheral blood of these patients treated with the IL-2. And I think most strikingly, and I think in the long term most interestingly, is that we repaired the STAT5 defect that I had shown you exists in type 1 diabetes. Remembering that the amount of phosphorylation of STAT5 that occurs with IL-2 in the type 1s is significantly lower than the healthy controls. Here, the treatment with IL-2 reversed that, and these now phosphorylated STAT5 much more like they phosphorylated in the healthy controls. But what was even more striking is if you looked 1 year out, after you'd already stopped treatment for most of that time, they still had better signaling through STAT5, suggesting that there was some permanent change in the cells exposed to this IL-2. And this just looks at each individual patient to show you this data where the majority of them had low STAT5 phosphorylation leading to high STAT5 phosphorylation at the end of the trial. So that all looked really good. The problem was we still saw increases in NK cells. We saw increases of IL-17 producing cells. We saw eosinophilia. And actually, the amount of insulin produced went down over the first couple months. It eventually came back. So we haven't really figured this out yet. So we know that IL-2 is a good growth factor for Tregs, but the challenge is how to use it in a way that doesn't also activate the pathogenic or potentially pathogenic immune response. And whether it's the use of mutants of IL-2 that only act down the STAT5 pathway and not down the PI3 kinase pathway, or it's some altered approach with other drugs, we don't know. But that's sort of what we're doing now, is trying to work on different ways to take advantage of IL-2. Another way we've tried to fix Tregs is, and you've already heard that I've been working on this for a lot of years, is to use anti-CD3 monoclonal antibodies. And this is now going back some time in the mid-90s, where we showed that if you could treat NOD mice with a short dose of anti-CD3, you could reverse diabetes in the majority of the animals. And there were a variety of reasons why that happened. And you cleared the islets. But most importantly, over time, the majority of these animals stayed insulin free, didn't need to inject insulin. And in fact, Tregs came into the tissue and protected the tissue from further damage. And this long-term effect was Treg dependent, was TGF-beta dependent. PD-1 is a functional molecule on the Tregs in this setting. And you can see here these islets started making insulin again. So even though they were there here, they weren't making insulin. So anti-CD3 is another approach that we've taken to try to boost Tregs. And why do we think this might work? Well, the anti-CD3 has a dual effect. On the one hand, it's very good at eliminating effector T cells. So this is just a DAPI stain looking what happens when you treat a mouse and look at the inflammatory site. And you can see a significant loss based on DAPI of activated T cells in this setting. At the same time, when you look in other places like in the gut, and you see that the anti-CD3 has actually increased the relative number of IL-10 producing cells and FOXP3 positive cells in these tissues, again suggesting this imbalance that we get with anti-CD3, that it eliminates effectors while boosting Tregs. And there have been a number of studies that have been done to show this. And so we think now that the anti-CD3 really is a potent shifter of the balance of an effector versus T response. And I'm not going to go into all the specifics here. It's work published with Kevin Herold showing how the anti-CD3 boosts that. So we've been in clinical trials with this antibody as well. Here's the antibody here. It's called teplizumab. It's mutated in the Fc portion so that you don't get cytokine storm by cross-linking the T cell receptor. So this is only a bivalently binding antibody. And we have found in multiple trials now, probably 5 trials, that the patients treated with the drug preserve their insulin-producing capability and require less insulin than the control group, suggesting that the drug actually does change the progression of the disease. We've looked in these patients and seen an increase in IL-10 producing cells, an increase in FOXP3 positive cells in this setting. And this is an improvement of about 88% and a delay by 16 months in the diminution of insulin production. But I think what's most important, because the results I showed before are okay, but since the insulin eventually goes down, I mean, you really haven't done much but prolong ultimately disease. But I think what's most exciting is some recent work that Kevin's done looking at patients from this trial and following them on the basis of, again, these are two parallel curves. They look different because they're plotted differently, but basically said, is there anything different from the patients that preserved insulin well versus those that really lost insulin at the same rate as the controls? So these we call non-responders, and these we call responders. And now, if you look at the responders, they've actually maintained insulin production well more than 2 years now, compared to the non-responders, which really lose insulin production at about the same rate as an untreated control. And we're now asking the question is, what is it that we can identify in these responders, some biomarker, so we can tell who's going to be the responder and non-responder? And what's interesting is at first blush, it looks like that the biggest thing that identifies these two individual populations is what their level of insulin production is before we start the trial. Those patients that need less insulin or have higher hemoglobin A1C, which is a marker of glucose dysregulation, hyperglycemia, those patients don't do nearly as well as the patients that start out with lower hemoglobin A1Cs and higher insulin production, less insulin requirements, suggesting that in some of these patients, the islets are too far gone and you can't save them even if you block the immune response. But if you can find the patients that still have good insulin function, that these patients might respond much better to the drug. So to summarize then the pharmaceutical or biologic approach to trying to do this, and I guess this is summarizing a whole bunch of stuff, we think that there's a future for using IL-2, maybe in combination with other drugs like anti-CD3 or rapamycin, PI3 kinase inhibitors, or thymoglobulin. So can we bypass, in a sense, the need for a drug like IL-2 or anti-CD3 by just going after the Tregs themselves? And can we, in fact, identify them, isolate them, and use those as therapeutics? So over the last 4 or 5 years, my lab's been involved in all of these areas to try to see if we can, in fact, develop Tregs as a therapeutic for use in autoimmune disease and organ transplantation. And I'm going to focus on 2 approaches today. The polyclonal Treg approach, which the FDA requires as the first in man, because you want to make sure they're safe, and a donor-specific approach that Kizi Tong and myself are doing collaboratively in organ transplantation. We also have some studies going on with antigen-specific Tregs, and in fact have been putting T-cell receptors using lentivirus into Tregs for potential therapies down in the future. So what are we doing in the polyclonal Treg field? Well, the first thing we needed to do was demonstrate that putting Tregs in works, and so we started with the animal models. And again, this is the NOD mouse, and if you take Tregs and transfer them into the NOD mouse, you can suppress pathogenic responses here, looking at gamma interferon. You can block disease progression. Importantly, and for future studies, the antigen-specific Tregs work the best. And most importantly for the clinical trials, the Tregs can actually reverse diabetes once it's already occurred. So we said, okay, maybe we can do this in human. So the first thing we needed to do was to isolate the cells. And so we found that using a combination of CD4, CD25, which I already told you about, and low levels of IL-7 receptor expression allowed us to isolate a very purified population of Tregs, here looking at FOXP3 expression on those cells. We could then use a combination in this. We used beads, anti-CD3 and anti-CD28 coated beads plus IL-2 to show that we could expand these cells over 2 weeks, anywhere from about 500-fold to up to 3,000-fold in this setting. And the cells that grow and come out the other end still remain very high for FOXP3 here based on staining that you can see here. And it's a lot of cells you can grow up this way. In fact, 5 billion cells represents close to about 10% of the Tregs that are probably in the body. And if you can do this in an antigen-specific way, you can imagine how much you could enhance it. These cells suppress very well. Here's looking at an in vitro suppressor assay. And they retain the demethylated FOXP3, as I said, the marker of a bona fide Treg, when compared to activated human T cells, which sometimes turn on FOXP3, but it's never demethylated. And I think most importantly, from studies I talked to you about earlier, is that the STAT5 phosphorylation of these cells is repaired, like it was when you treated with IL-2 in vivo. So if you compare again controls versus the type 1 diabetics, which have reduced phosphostat5 with IL-2 engagement, these cells, after 2 weeks in culture now, phosphorylate STAT5 almost as well as healthy control populations. And you preferentially are growing out the RA cells in this culture, if anyone cares, and we have less than 5% of the cells make interferon or IL-17. And they suppress better in vitro. So we're now in clinic with these. We started on a dose escalation trial in type 1 diabetics, taking patients from 3 months to 24 months after diagnosis. We started in adults for safety reasons, and we're looking at safety as a primary outcome. We completed the first 2 doses, cohorts in this. This is 40 million cells. We still have a ways to go. And starting in the fall, we're going to go up to 320 million cells, and then hopefully by the end of the year be up to 2.6 billion cells that we're putting back into these patients. So far, the grow-ups have been very good. The cells look very good. When you look at them, we're not getting much contamination or loss of FOXP3 expression. And although we haven't seen any changes in Tregs, we don't expect to see that with so few numbers putting in, at least we know we can monitor these things in real time and see if there's any change in the Tregs. And in the next cohort, we're actually going to be labeling the cells with heavy glucose that we inject so we can monitor the cells we've injected by isotope analysis. So our goals at this point is to complete the final 2 cohorts and get the tracking going and get the phase 2 trial started. Again, the last point is that the C-peptide hasn't changed, so that's good news so far. So the last few minutes, I want to talk about the work that Tong and I have been doing looking at donor-specific Tregs, because ultimately this is really what you want to do. Why? You'll increase the specific activity, and at the same time, you'll reduce the chance for non-specific suppressor effects that you might see if you put in polyclonal Tregs. And so Tong worked it out in the mouse first, where she found that if you depleted alloreactive cells using Cytoxan, following donor-specific antigen, and added in donor-specific Tregs, you could get very long prolongation of, in this case, we're looking at islet allografts in these animals. So we knew that a combination of a depletion of some effector cells and Tregs were very good. The donor-specific working much better than the polyclonal Tregs. Okay, so the protocol that we'll ultimately use is going to be a combination of this debulking and donor-specific Tregs. So how do we grow up donor-specific Tregs in this setting? Well, instead of using anti-CD3 and anti-CD28 beads, instead we've used activated B cells. Because activated B cells, here being activated by CD40 ligand activation through CD40, express very high levels of HLA-DR and the co-stimulatory molecules CD80 and CD86, which Tregs need to grow. And if you actually look after growing against these B-cell blasts versus control, you can see that the majority of Tregs actually respond to the allografts in a mixed lymphocyte reaction compared to the polyclonal Tregs. This is dilution of CFSC, which is a readout of a cell that's responding to the alloantigen. And so we now have a protocol we put together for human in which we stimulate for 9 days with these B cells that are expressing the alloantigen, and then follow up with beads to get full expansion of the cells, again out hundreds of fold. They maintain their FOXP3, they maintain their demethylated FOXP3. They respond really effectively well to the alloantigen compared to the polyclonal, which hardly respond to the alloantigen at all. They even respond better to the alloantigen than they do to anti-CD3 and anti-CD28. So we can get a very specific population that suppress at least 10-fold, maybe 20-fold better in an in vitro assay. And so we're in the process of getting ready for our first clinical trial in liver transplant, working with Sandy Fang at UCSF, in which we're going to take liver transplant patients and we're going to convert them over to what we call Treg-friendly immunosuppression, thymoglobulin and rapamycin. And then at 90 days or so, we're going to infuse these alloantigen-specific Tregs and follow up and ultimately see if we can withdraw these patients from immunosuppressive drugs. We have actually 4 trials planned, one here with a European group in kidney transplantation, and then a number of other trials. And so we hope then that we'll be able to see over the next few years whether this is a therapy that can work. So let me just summarize what I tried to tell you today, that Tregs are indeed essential for immune homeostasis and can be defective in the autoimmunity setting and may in fact also lose FOXP3 in infectious disease settings, which may be good because you want the effector cells to respond there. That Tregs have been shown to be effective in multiple mouse and humanized mouse models, but their instability has got to be taken care of, and a lot of that is due to an inability to respond appropriately to IL-2. That not only IL-2 but TCR CD28 signals are critical, and microRNAs, P10, and almost undoubtedly other downstream signals are going to be targets that the inflammatory setting are going to compromise. Finally, that Tregs and IL-2 therapy hold some promise, at least in animal models, and we hope that we'll be able to demonstrate in humans too, at being able to reverse autoimmunity, lead to tolerance, and do this while being able to manage conventional immune responses and Treg stability. And we hope that ultimately we'll be moving to an antigen-specific population, first with the alloantigen and eventually to populations that see autoantigens like insulin or myelin basic protein, and that we have multiple trials underway. So let me end then by thanking the people who did the work. In my laboratory, Sam, Mark, and Mahesh have been involved in the stability studies I talked about. Lucas, Jonathan, and Dimitri in the microRNA work. Michael DuPage in the P10 work. Joe was involved in developing the reporter mouse that I talked about. I mentioned Larry Turka. We've done a lot of the work with him on the P10, and Jane Buckner's group at Benaroya did a lot of the human STAT5 work that I talked about, not just the published work, but our own work looking in the clinical trials. And we've got funding from National Institutes of Health as well as the JDRF. And then it would be really unfair not to also thank the clinical team. These studies don't get done without an enormous amount of work by a lot of people. The clinicians who've been very involved, people like Amy Putnam, who spends her life in the GMP facility growing up these cells, Mike Lee, who sorts them, Wei Hong, who analyzes these cells, various groups at UCSF that handle all the clinical work, and then folks in the Department of Surgery who've been very involved in the allo work that I talked about. So I'll stop there. Thank you.